In a case study 2, it's a strain engineering. So another client was interested to discover mutations in a gene of their interest in a few bacterial states. For this case study, we choose RIB-F gene and 4 E. coli strains. So according to them, this typical challenge are the lack of user-friendly visualization tools, or even they are available, they are like standalone. So you just can't link them in a typical workflow. Now, to discover the mutations, a scientist needs several files, such as BAM, VCF, GFF3, and more. And it will be a labor-intensive process to collect all these different kind of files and process them and get the information of the mutations. But even say the scientists settle for command line based querying and mining of the data with the help of a bioinformation, their availability is another big challenge, because the bioinformation are busy and they're supporting multiple other projects in the R&D team. So that are the challenges you have to overcome to do this kind of work. As a quick note, for this case study, we are using data from NCBI and not data from any of our clients. So as I mentioned earlier, in this typical workflow, you require to have the BAM, GFF3, VCF, which are generated during the assembly and annotation steps. So as a bioinformatician, I know that collecting different files and then processing them is often very troublesome and challenging steps to go further and take out the meaningful information. So here, Loci helped them to process all these steps. Now, to proceed with the project goal, click on the genomes on the navigation bar after login, and we downloaded the whole genome sequencing data of representative multiple organisms such as bacteria, archaea, plants, protozoa, virus, fungi from the NCBI, and arranged them into two major genome collections, namely public and private. So we name them as public and private for better understanding. Here is a flexibility how you want to arrange. So we arrange them like 3 versus 3 into private and public. This page is customizable, and as I mentioned, so you can store data according to your need and the projects or the organisms. Now to proceed further with the project goal, click on the genomes on the navigation bar, which is symbolized by the hand symbol. Now after coming to this page, you'll see the search bar. So you search for the reference sequence data of E. coli strain that was used and click search. Now since JBrowse is already integrated on Loci, so no need to install any other JBrowse standalone applications or any other visualization tools. You just click the browse to visualize the E. coli reference genome through the integrated JBrowse. And you can easily visualize the nucleotide sequence and amino acid sequences. To add additional track, click on the track button on the menu. Now add annotation file in GFF3, reference SNP variant file, and BAM files of interested E. coli strains by selecting those files from stored location. Now you can add these tracks one by one or all together. Excuse me. Now adding all these tracks can be done by the bioinformaticians themselves, or give the file locations to scientists to add themselves, because it is very straightforward by adding tracks. Now on this browser, you have reference sequence, annotation file, reference SNP variants file, and the BAM file. And you can change the order however way you want to see them. Now to search for RIB-F gene, which is our interest in this big pile of genes, simply type the RIB-F gene here in the box and click go. The whole page of the browser with all the files will be zoomed in to RIB-F gene. And to know more about this gene, click on the RIB-F gene. So here are all these relevant information about the RIB-F gene. You can download the sequence by clicking on this button, which is symbolized by the hand symbol. And there are two reference SNPs in RIB gene. To learn more about this SNP, you can click on the SNP itself, and the user can easily visualize the variations at genomic region at RIB gene. So here, if you look deeply, you can see there each vertical strip of color represents a nucleotide difference from the reference. In this case study, the user can detect 8 genomic regions that are varying from the reference. So with respect to reference SNPs, strain 1 is different from rest of the strain. To correlate the mutations with amino acid change, zoom in the reference sequence region. So in this case, strain 1, which has UGA at this region, which translates into stop codon. And however, the other strain have GGA that codes for the glycine amino acid. These results can be shared with your colleagues by clicking on the share button and send it through either email or instant messaging app. So in this case study, we discovered that there are 8 mutations in the RIB gene, which compared against the multiple strain. One of the mutations clearly differentiates strain 1 from the other strain. Also, scientists could quickly correlate the mutations at the nucleotide level to amino acid and thus protein level, and share the whole information with the colleagues. So in this example, we use FASTA, GFF3, and BAM and BCA. But if a scientist want to analyze with the big bed and big weak fields, they can also do that. So in this case study, we showcase discovery of mutations in one gene. Imagine if you have like multiple genes and how quickly mutations can be discovered. So Loci make this complex mutation discovery easy for scientists under, say, 5 minutes.